was fast enough. It's alright. Okay. Thank you for bearing with me on that one. A few thoughts. I do not fit in Dr. Enos's lab coat, so please don't tell her that I put it on. This ought to be fun. There we go. All right, how's everyone doing? This is it, last week. Yeah, I think I'll have some kind of more lecture-related material tomorrow and Wednesday. But today, I wanted to just post kind of another video lecture in the series of the McGregor et al. Paper. And this will be probably not the last longer one. I'll maybe post a shorter one on Friday to just kind of sum things up. What I want to do two things in this one. I wanted to do a bit of a figure 2 recap. I had a chance to go through at least those responses that have been posted, and then a little bit of a figure 3 intro, and then I'll present the kind of 2 discussion questions that I want you to work on this week. So for this figure 2 recap, kind of one of the ways that I structured the questions for this one was to try and get you to develop a habit. And when you start reading literature and you get into a figure, I want you to develop the habit of being able to ask, what is the point of this figure? Okay, so clearly the authors structured their paper in this way for a reason. Okay, so they're trying to sort of communicate some sort of big point to you, and they're trying to do it in a logical way that you can understand it. It presents both the rationale behind why they did it, what they did, what their results were, and how that informs their conclusion, right? So I want you to get in the habit when you get in front of any kind of a big complex figure to kind of approach this in 4 main parts. The first part is to basically do that first question that I had you do, which is, what question is this figure trying to answer? What is the broad research question behind this particular figure? Then you can follow that up with, what are the data? Okay, so built into that is sort of an idea of what are the experiments they're doing, what are the data they're producing, and can actually sort of get a handle on what are the real results they're presenting in this particular data figure. What are the data? And then from that, you can look and look and see what are the authors trying. What's their conclusion from this figure? Okay, and then then comes the last most important part in any kind of a data figure. And any time you're analyzing or reading primary literature, you have to sort of judge on your own whether or not you think the authors did a good job. Okay, so that last question then is, does the data that the authors show in that figure support their conclusion? Okay, so this is a big skill in reading literature, and it takes a long time to develop. And I think this is important not just for reading scientific literature, but I think this is a pretty good big life skill. Okay, do the data that the data the authors present support their conclusion? Okay, are they making as good of an evidence-based conclusion as you think they can? All right, so those are kind of the big skills I want you to try and develop as you read literature, and maybe kind of pull some of the more universal things out of that as you kind of move on past scientific literature. All right, so figure 2, I'm going to kind of go through and actually provide some examples of things that I like the way people answered the questions in figure 2. So the first question was about that broad research goal. Okay, and so Jacob says something to the extent of, what regulatory regions specifically constituted this difference in expression? Okay, Kylie, where are the genetic differences that caused reduced expression of the SVB gene in Drosophila seychellea? Okay, I like that. I'm really putting together a couple different points here. Hamid, asking the question, which enhancers are responsible for the expression of the SVB gene in the epidermis of Drosophila? Again, I like that one, kind of getting this idea that we're parsing down that promoter in different spots. Victoria, what enhancers are causing SVB to be expressed differently between these 2 species? I like that one because it brings up that contrast between Drosophila melanogaster and Drosophila seychellea. Elena, what enhancers are responsible for the SVB expression, and how have these enhancers evolved in Drosophila seychellea? Going to kind of react, bring up this bigger idea in the paper, this idea of the evolution of those enhancers over time. And then Kayla, what enhancer regions regulate the SVB gene in Drosophila seychellea and Drosophila melanogaster, and how do these regions compare between the 2 species? Again, a nice comparison between melanogaster and seychellea really kind of encapsulating what it was they were trying to do. Okay, so I like all of those, and I think all of those really come pretty close to sort of encapsulating really the totality of what they're trying to ask in Figure 2. Okay, they've identified these. They identified in Figure 1 that the expression pattern was different. In Figure 2, they actually went into that upstream region and try and figure out to find different regions within those, and different enhancer regions within that kind of greater cis-regulatory region of shaven baby, and find out whether or not there are actually distinct functional differences between Drosophila seychellia and Drosophila melanogaster. Okay, and then on toward Figure 2, the conclusion. So what was the conclusions the authors drew from this one? Okay, so there's, again, I'll go through some examples of people's answers that I liked in this one. Natalie, they found that these enhancers would promote transcription of the SVB gene differently, okay, getting to some idea that there's some difference in this expression pattern. Let's see, Jesse, 3 enhancer regions, distal, medial, and proximal, have evolved in Drosophila seychellia that are responsible for the differences in the expression of SVB between Drosophila seychellia and Drosophila melanogaster. Okay, kind of getting back to this idea of the evolution of these cis-regulatory regions. Robert, the regions in which these enhancers promote SVB expression differs between species. Okay. Brianna, these regions, however, regulate the gene differently in the 2 species, causing different trichome patterns between the species. Okay, I like this one because it kind of brings things back to the sort of morphological difference, the trichomes themselves. Okay. Preston, the main conclusions the authors draw from Figure 2 is that there are 3 separate enhancer sequences that differentiate the expression of SVP between Drosophila melanogaster and Drosophila seychellia. Okay, this one's kind of, I like this one because it just sort of takes that question and sort of makes it into a statement. You can often play this game in individual figures. So there's going to be a question, and then the conclusion of the figure is really just a statement of that question. Oftentimes the answer is yes. In this case, this is essentially what Preston's getting at here. Okay, and the last one, Courtney. Broadly, love that term. Broadly, the researchers found that 3 enhancers, labeled dorsal, median, and proximal, were responsible for the expression of the SVB gene in the epidermis, and these enhancers turned on SVB expression differently in Drosophila melanogaster and Drosophila seychellia. Good, another one I think that encapsulates a lot of stuff that's going on in that particular figure. Okay. The third question was this statement rephrase, and this one was tough, and I think it was tough in part because after kind of going through the author's sort of writing, I think what I ultimately decided is that I don't like the way they wrote that phrase. I think it's pretty confusing. I think it's overly jargony, even for a paper that's in the journal we're reading, Nature. That's a pretty kind of very science-heavy journal, but I still think that statement itself was a little bit too jargony. Okay, so there kind of one key term that people got hung up on was this idea of cell autonomous. Okay, and so this is actually pretty, well, how do I want to put this? It's a pretty thick term in terms of its use in cell biology and development specifically. Okay, so I think a lot of you sort of correctly got to this idea that cell autonomous means sort of in the same cell. Okay, so it helps to envision this with an actual diagram of what's going on here. So this is the cell we're talking about, and let's say this has a nucleus here. So what they're saying is that SVB, which is a transcription factor, I think that's the key here, is able to then produce or transcribe the mRNA for miniature. Okay, and so this idea of it being cell autonomous means that SVB is doing this. The miniature is being produced from the same cell in which SVB is acting. Okay, so it doesn't make a whole lot of sense really in this context, where oftentimes, this is why it was confusing to see this, oftentimes people refer to cell autonomous or cell non-autonomous when they're worried about neighboring cells. Okay, so in this case, it's entirely possible, and this does happen in multicellular organisms. We've got another cell, kind of a neighboring cell to this one. Let's say that SVB then regulated some some product, we'll call this X, and this is actually a secreted protein that can bind a receptor on the neighboring cell, and this can then send a cell into this nucleus, which then ends up transcribing miniature. Okay, it's pretty complicated. There are situations where this happens, especially during development. And this would be a cell non-autonomous effect, okay? So this would be this SVB in this cell affecting transcription of miniature in this cell here. That would be cell non-autonomous, okay? I found this statement to be somewhat confusing, especially given the prior research that showed that SVB directly regulated the transcription of miniature. That was that figure, that reference that I had you look up, reference number 13. That paper showed that SVB directly regulates transcription of miniature. So I felt like they didn't need to use this term. I think it was overly complicated. They could have just said something more simple. They really honed in on this idea that SVB is a transcription factor. And so a couple of you actually got there, 2 of you. Carl said that miniature is a product of a gene that is targeted by the SVB transcription factor. Simple, straightforward, to the point. Also Laura, shaven baby encodes a transcription factor that regulates the expression of several genes, including miniature. Okay, I think either one of those statements was a better statement than what the authors used in this figure. Okay, so that's sort of a broad lesson, and even when you're writing a very scientific paper for a scientific audience, you can always sort of pare down on the jargon. Okay, and then figure 1b, this was sort of the overall conclusion of the figure 1b that I had you look up in reference number 13, and I think everybody did a fantastic job with this one. So good job in sort of reapplying your knowledge about in situ hybridization works. Nice job in finding the paper. Sometimes that can be difficult. And so I think there's one. So Hunter, I liked his sort of overall conclusion. He said, however, when the SVB gene was inactivated, trichomes were not present and miniature was not expressed. This suggests that the SVB gene regulates the transcription of miniature. Okay, so I like the way he kind of talked about both the results in the figure and then took those results and put them together to that final suggestion statement. Okay, and of course that figure released only suggested that SVB regulates miniature. They went on later in that paper to show much more clearly that SVB does directly regulate the transcription of miniature. Okay. All right, so that's kind of a recap of figure 2. Again, thanks for everybody for putting in that response. If you're watching this video and you haven't put in a response, please do. I'm not trying to be a stickler for deadlines on this one. I think pretty much everybody, if you put something down, I would say definitely everybody, if you put something down, it looked like you kind of made an effort at answering all 4 of those questions. You got all 4 of those points. Okay, so at this point, if you haven't put a response in there, please do. The main thing is I want you to kind of go through and read the paper. All right, so where are we at so far in our story? So far, we've done a lot of comparison of Drosophila melanogaster versus Drosophila seychellia, with the exception of figure 1b. They kind of threw in a couple other fly species in there. We're going to come back to one of those here in figure 3. And so they clearly showed there's a difference in trichome pattern between seychellia and melanogaster. There's clearly a difference in SVB, or shaven baby expression, between melanogaster and seychellia. That was all really established in figure 1. They went on then in figure 2 then to dive into why there's a difference in expression between difference in shaven baby expression between seychellia and melanogaster. And they did that by focusing on these different enhancer regions, proximal, medial, distal, and making transcriptional reporter constructs and putting those back into melanogaster and showing that these enhancers actually regulate gene expression differently. Okay, suggesting there's something different about these enhancers between seychellia and melanogaster that leads to this difference in shaven baby expression, which therefore then leads to this difference in trichome formation. Okay, so let's get just to figure 3. Okay, and so the big question driving figure 3 is, how do these differences evolve? Okay, so we're getting back to that big picture. I think a lot of you picked up on it when we talked about summarizing some key aspects of the introduction, and especially in terms of the broad research goal of this entire paper. Really, the big kind of goal of this paper is to dive into, again, the genetic mechanisms underlying the evolution of a trait, a morphological trait. Okay, so just a broad comment on this. Any kind of experiment or study or research project that's kind of looking at evolutionary mechanisms is almost by definition going to be very hard, right? Not impossible. It can be done. Don't let anyone tell you you can't study evolution. You very much can, and you can actually study the mechanisms of evolution. You can show how evolution occurs. I think there's plenty of examples in bacteria. Sort of the evolution of resistance is a really cool kind of more fast timescale type of experiment. But when it comes to understanding how different things evolved over time, these are really tough experiments. These are things that have already happened. So you're often looking for kind of hints in sort of current biology, kind of, I would say current by those things that are still existing. What exists in current biology that can inform us of that distant past, of where those changes occurred. So a lot of that can be read through DNA sequence, and that's essentially where this thing is going. Maybe not in this paper, but overall, you can kind of think that there are going to be these sequence differences between Seychelles and melanogaster, and eventually they can kind of come up with what those are. So they're tough experiments, but they can be done, right? So kind of a more specific question kind of underlying this one is, what are the genetic differences in shaven baby that lead to the trait difference? And again, our trait here is trichomes versus no trichomes. Okay. So they got at this a bit in figure 2, and they're going to continue on with this in figure 3. Okay, so let's kind of hone in on these differences, these changes. Okay. And a little bit about kind of the big picture of this paper and this idea that they were trying to contrast large, abrupt changes. They're trying to contrast that with small effect changes at multiple locations. This is kind of when they set up their introduction, they set up this kind of big either-or situation. So previous research has suggested that this is how evolution occurs, or this is how evolution occurs, but this doesn't quite explain how we see these kind of large abrupt changes. And so in the case of this paper, the large abrupt change that they're looking at is actually the change in SVB expression. Okay, it may not seem like much. That's actually a pretty large change within sort of speciation, to go from expressing a gene to no longer expressing it. And maybe it's not quite that binary, but that's a pretty large abrupt change, right? And so what they're trying to do is show, their argument at least, let's see here, is that a combined small effect change at 3 locations is really what contributes to this large abrupt change. Okay, that's their argument in this paper. Okay, so while up to this point in figure 2, they've identified these sort of 3 different enhancer regions that seem to differentially regulate SVB or shaven baby expression, they haven't quite sort of put this argument together yet. And I think that's what they're trying to get at in figure 3. Okay. So, a couple notes about figure 3, because this one's thick. There's a lot going on here, and again, just like all the other figures, I do not expect you to come out of this understanding all the sort of finer detail out of this, and even I don't understand a lot of the finer details of this figure. So my kind of advice for you is do your best to understand roughly what they did. Okay, so try not to get bogged down by a lot of the details. Try and just kind of think big picture about what they're doing in this particular figure. And so my hint for this one is that they mated Drosophila seychellia with another one of the species they actually showed in figure 1b. This was Drosophila, I want to call it Mauritiana. I may not be pronouncing that right, but it's Mauritiana, Mauritiana. And so this one, Mauritiana, has a trichome pattern similar to melanogaster. Okay, so I'm not quite sure why they did this experiment mating Seychellia with Mauritiana. It may just have to be, it may have to do with some sort of just feasibility issue, but this was kind of broadly how they started this thing off, by mating flies from Seychellia flies with Mauritiana flies, okay? And after this, then they analyzed recombinant progeny. Underline that because that's a term you should understand, or at least recognize. Okay, so they're looking for crossing over. Okay, so this is kind of what they're looking to happen in figure 3. They're looking for this. Okay, so what they're doing is they're focusing on recombination events that occur within the upstream regulatory region Okay, by this I mean that region that includes all the enhancers. It's the upstream region of a gene that contains a lot of regulatory elements. Okay, so they're looking for recombination within the upstream regulatory region of SVB. And that then change trichome patterns. Okay. So again, it's a pretty kind of heavy figure, and I don't expect you to fully understand the method that they do in this one. But I do want you to try and get as much of the kind of broad points out of it as you can. All right, so I'm not going to go into too much depth on this one, but I am going to ask you 2 different questions that I've already posted on Katie about figure 3. So after you give yourself a chance to go through figure 3, try and extract some information out of it, I want you to answer these 2 questions. Okay, so the first question has to do with the statement they say, or the phrase they use that says native genomic locations. Okay. So the authors make a point of saying that they want to test the function of the shaven baby enhancers, quote unquote, within their native genomic locations. That's their quote, their phrase. So my question to you is, what do you think they mean by this statement, native genomic locations? And how does this differ from what they did in figure 2? Okay, the second part of this question then is, why do you think that it's important to study the function of the enhancers in this context? Again, this context meaning native genomic locations, right? Okay, then question number 2, again, a 2-part question. Okay, the first one, in what way do the results in figure 3 support their conclusion that 3 separate genetic changes led to the difference in shaven baby expression and therefore trichome formation in Drosophila seychellia? Okay, so that's their kind of broad conclusion, that they're a combination of 3 small changes led up to this large abrupt change. What parts of figure 3 support that conclusion? Okay, and then the second part of question 2, what would the authors have expected to see in the results of the experiment in figure 3 if the Drosophila seychellia trichome pattern resulted from the evolution of a single site? Okay, so you want to pay attention. I think there's a sentence at the beginning of one of the paragraphs, kind of as they're going through the data in figure 3, that you really want to hone in on to answer that particular question. So just kind of playing the game of what would they have expected to see if their hypothesis were wrong, if there weren't 3 separate changes, if in fact there was just a single change that led to this abrupt change in SVB gene expression. Okay? That's it for the discussion questions. Those are due Friday, this coming Friday, May 15th at 11 p.m. Central Time. This is worth 4 points. Yes, that's all I've got for this one. So have fun digging into figure 3. Again, don't worry if some of it doesn't, or most of it doesn't make sense. Just do what you can to try and get some information out of it. All right, look for a couple videos tomorrow and on Wednesday, continuing along the lines of genetic engineering. And I think we'll leave it at that. All right, see ya.